Now it's a pleasure to introduce my friend and colleague, Dr. George Dangas, who is Associate Editor for JACC and Director of Cardiovascular Innovation at Mount Sinai Health System here in New York. And he's going to talk about designing and conducting large international multi-center randomized controlled trials.
Thank you, Dr. Fuster, and also many thanks to Professor Gilles Montalesco, who organized this great session from the other side of the Atlantic. Following this great and concise overview of JACC, let's try to go and look a little bit more closer into the conduct of clinical trials and what ideas could help international researchers focus more and get high-impact studies and subsequent papers going ahead. The study design of clinical research has many stages and have something to do from the conception of the idea into various stages, maybe some retrospective chart reviews, often in the basements of the hospitals where the medical records used to be, some comparisons with historical controls, perhaps under some exploratory abstract submissions and presentations, ultimately to a discussion of a single center study and go ahead to get multi-center interest, often international, to a strongest level of evidence, which is a large randomized clinical trials with multiple centers. The most important thing ultimately is to figure out what is a control group characteristic and what is a new intervention or therapy that you're trying to bring along. I see a lot of confusion on many levels on the statistics and what the statistics are and the statisticians, and I'm sure there's a lot of work there. As you will see in my slides, I will have nothing about statistics because in the end, the quality is primarily on what we're going to talk today, which is how you configure your control group, your treatment group, and what you think is important. Because when you face into something that perhaps didn't go at many places, either in presentations or publications, you will see some of the situations that have arisen have nothing to do with statistics, have everything to do with perhaps the control selection wasn't so clear. And maybe there was disagreement about this was an appropriate control group. This means you're comparing against something that not everybody agrees is a current standard of care. Perhaps your treatment assumptions were huge. Many people are going to say, what kind of study is that? You have unrealistic assumptions. You're coming in trying to reduce mortality by 90%, for example. No matter what your result is, that's going to be criticized in the end. And this is a clinical decision up front. How you define your endpoints is also important. We talk about MI, stroke, and many other endpoints, bleeding perhaps, and there's a drive these days that the clinical trials around the world to be associated with a standardized definitions. So if you try to do a new study and all of a sudden you introduce your own definition of MI or stroke, well, that's a decision you are making up front that may haunt you in the back end of the study because others are going to say, you know what, that's not consistent. That's not the way we define MI, and therefore you're not going to be able to get through the reviewers very successfully. Or you may have an issue on how practically the study is done. Perhaps the protocol is great, but so complex that nobody could follow it. You have a lot of violations and deviations and so forth. You have many people crossing over or stopping the study drug or whatever. And all these situations may, in the end of the day, introduce bias. They make the study results not so clean. The limitations of a clinical trials also is the time it takes to completion. And we've seen that a lot. A lot of frequent criticism in JACC is, you know what, we had this great study, sounds good, but you know what, why it took them 6 or 7 years to complete the study? And by this time, the control and the assessments of the standard of care have changed. So study may not be so relevant, although in the end of the day, it was conducted okay, just took too long. Or perhaps the study treatments that you're proposing have been surpassed in some other way. Perhaps you saw some recent results with ways to do mitral valve repair. This affected immediately many other studies that were proposing or actually conducting research on mitral valve replacement. And there was a question whether either their control or their active arm should be adjusted accordingly. So you can imagine if your study takes too long, many of these things are just going to appear and you're going to have to deal with them somehow. And in the end of the day, in order for your study to be believable, to affect the guidelines, be highly publicized and discussed, the key factor is it has to be accepted somehow as generalizable. The variability around the results or the way that the patient selection to get in the study was executed and planned will affect on how broad the applicability of the study, of your study is. And in the end of the day, a study may be great, it may reflect some results, but if it ends up reflecting the results only applicable in this narrow population studied, that's kind of a limited scope. If somehow it's generalizable to a greater population that may impact health through the affect perhaps the guidelines, etc., that's even a study we all want and we desire. What is the role for the observational studies, and how can we kind of learn from that the way we can affect a conduct of a randomized trial? The observational studies have an inherent bias that you don't know exactly what was the decision making for the given therapy. And nothing can get that away from what I'll be talking to. But on the other hand, they have great numbers. And again, nothing can take that away. Why? Because you no longer deal with a narrow population that specifically consented to be subjected to a randomized trial after so many screenings, several family meetings, many discussions with doctors, and a lot of scrutiny from everyone. So you really try to replace the unknown decision for a selection of a certain therapy with a huge number you can put behind a much easier way in an observational trial. Can they overcome their limitation? Let's not be overachieving here. A non-randomized trial will never be able to eliminate, even by propensity matching, the bias of why exactly was this type of therapy selected for this patient. So I think we should forget about eliminating completely. We have to minimize it. Propensity matching perhaps is one of these many ways, multivariate analysis, but you can capitalize on the strengths instead by making it easy to enroll, very fast enrolling. Therefore, you get away from the difficulties I outlined before regarding the control group being overcome by new developments, etc. And you end up with a highly generalizable result in a very huge sample size. So you muscle out your limitations by boosting your strengths. If you also fail to do that, if you have an observational study that takes a long time and nobody's putting patients in, then obviously you cannot overcome your limitation. At the same time, you're not playing up your strengths either. Again, this slide deals with what I called before no way to know exactly why a certain therapy is selected. In a more academic term, this is called the unmeasured confounding, like something that I know as a doctor that assigns treatment A to treatment B, but it's not exactly captured reliably or clearly in any of the baseline characteristics. It's not the diabetes, it's not the smoking, it's not that. Is it perhaps a frailty? Is it just my preferences? And somewhere there, this exact information is unclear and becomes a so-called unmeasured confounder in the observational study. So in the end of the day, and I'm going to move to my next section very soon regarding the collaborative trials, we're going to see that in order to overcome every result, we have to also make sure that we have a great design and a great concept to study. The pros of the global study is that it can be very big and it can be very much generalizable because it includes a lot of different regulatory requirements, a lot of different standards, and a lot of care differences. When you plan the study, these are difficulties. After you've done it, they become pluses. because yes, they're going to give you a little difficult time to complete it, but once you complete it, then obviously taking account of all these factors becomes a plus. And international collaboration is very much well balanced between evidence-based, the clinical judgment, and the socioeconomic forces that obviously are going to be maximizing this type of trial that is not taking care of only one kind of population, but many. There are various studies regarding this type of studies. I don't think there is anything more to be said, but just what I said before, there's nothing here that boosts the mathematical statistical part into a driver's seat. Everything is about what you think is missing from clinical point of view, a clear understanding of the standard of care, and a clear development of an active treatment arm that addresses this problem. And obviously, there has to be, the first situation is that you have to be somewhat unhappy about the current level of state of a certain disease, situation, or other syndrome. There are many steps from the end to the finish, and you can see that there's quite a few to be done. And unless you have a very clear understanding of how you end up finishing, then you will have a lot of difficulty with each and every one of the steps. But with a more clear goal you have for the very end, you're going to have a very, very simpler way, and I didn't say easier, I just said simpler way to accomplish all these different steps. All right, regarding the operational issues, I think everybody who is somewhat involved in the clinical trials understands that the study design and the graft protocol and the circulation of it to a series of experts is extremely important, as well as extremely important to set right in the right way the checks and balances of your clinical trial, including the ethics committee and the various monitoring boards. You have to have a lot of account for any operational phases and not underestimate the finances and the different meetings. You have to be very clear of how the closeout is going to look as far as a process in order to not miss anything, at the same time not prolong the whole operation for very, very long. And I do think that in the end of the day, every I would say medical center in every country has figured out that in order to have successful research that is believable and generalizable, we have to go to national data repositories, comparative effectiveness research, and ultimately international collaborations for any of these situations. What I'm depicting here is an example that we know we use many devices. Electrophysiologic devices have unique patient identification. We always know which device belongs to each patient. Many other devices don't, although many doctors and patients think they do. Imagine if we get this type of activity going, what kind of incredible research we could be doing with a very, very much close identification of a certain device of a certain patient, and then you can try to take advantage of what's happening in the globally with this type of situation. I think I'll probably leave it at that. I think the incredible opportunities for device surveillance and this type of research all over the world is very important. However, we may get into that in the question and answer session a little bit better. Thank you for your attention.